Michael Blink.
It's my pleasure to speak to you today. And what I'll be talking about today is the multi-attribute method, which is a technique that brings mass spec into a GMP or quality control lab. So as you're aware, a monoclonal antibody drug is a large and complex macromolecule. You're talking about tens of thousands of atoms. And a modification at any one of these amino acids can have a profound impact on the safety and potency of the drug. So you can see some of the heterogeneity that can be implicit inside a MAB drug. You can have oxidations, you can have changes in the glycosylation, you can have glycation, you can have pyroglutamation, lysine clips, all of these. And some of them will have a small and negatable effect, and some of them will have a profound effect. And the idea here is that you want to be able to tie in how changes in safety and potency that are measurable by other means relate to the chemical structure of these drugs and increase the knowledge of how changes in process and in manufacturing affect the chemical structure. And what this enables you to do is start to develop a quality by design regime where you can actually begin to envision a control space where you say a change in pH at this phase changes the amount of isomerization at this phase, which changes the safety or the potency of the drug. And so now you can say, when we're doing manufacturing or when we're doing any kind of process validation, the operating conditions need to be kept in this range to get the drug molecule that we want. And mass spec is the ideal tool to do this because of the wealth of information that it provides. So in a typical characterization battery, you might perform all or at least some of these tests, right? So the techniques that are part of a conventional lot release strategy are shown on the right here. And so cation exchange coupled with UV will give you some of the information. So for instance, charge variance will give you C-terminal lysine assessment. Glycation, glycosylation can be achieved by Hillick. HCP ELISA will give you some idea of the amount of host cell protein there. You can get the identity from ID ELISA. But what you'll notice is that each of these tests is only giving you one or a few pieces of information, and that even if you did all of them, you're still not going to get all of the information that you could potentially access through a mass spec method. Now, mass spec method won't give you everything. You see that there are some gaps and some limitations. But because of the wealth of information that it provides, it not only is enabling you to replace some of these methods, but giving you a deeper understanding of the product. So it's about increasing the knowledge and safety of the drug itself. So the way that this workflow works is essentially we've been recommending about 3 micrograms of digest. You can see we're using the non-proprietary NISMAB here as an example. There's some high-resolution separation, so this is done using an AccuCore Vanquish C18. And then UHPLC. The Vanquish is ideally suited to this, especially the Horizon. It will go up to a 1500 bar. So this enables you to really take your pick of columns. And my colleague Stefan has actually come up with this really clever way of stringing several 25-centimeter columns together. And the Vanquish, because of the higher pressure limit, will be able to take it. You can see that there's a blanking gradient at the end. And then the actual work is done on any Thermo Exactive series. So this method, all of the details that I show you here are compatible with everything from the Exactive Plus, which is a really great entry-level, high-resolution instrument. It's very easy. It's only 15 minutes to calibrate, not that complicated, all the way up to the, does everything QEHF BioPharma, which is your all-in-one solution. And I'll briefly discuss. I don't want it to be too markety, but I'll briefly discuss why you might want one or the other. There's a discovery phase. This is done in BioPharma Finder. This answers the question, what should I be interested in? What should I be looking for? Somebody asked me a couple of weeks ago, find any sequence variants. This is a molecule that I'm not familiar with. I have no idea what sequence variants could be there or will be there. So I used BioPharma Finder to look at the peptide map and say, identify anything that's a sequence variant from the sequence that they provided. Once I know what that is, then I can actually do routine monitoring of those sequence variants in a GMP-compliant fashion using Chromeleon. And Chromeleon enables CFR 21 Part 11 compliance. It'll do all of the acquisition, the processing, and spit out some cool reports I'll show you. So for this workflow, we recommend kind of a gentle digest. This is denaturation with guanidine. This is based off of Amgen's protocol that they published in Analytical Chemistry in 2009, I think, by Da Ren. reduction with DTT, alkylation with iodoacetate, and then we desalt. And then the trypsin digestion is 37 degrees at 30 minutes. So it's very gentle. Because this is one of the questions that we always get. How do you make sure that the attributes that you're monitoring, say for instance like isomerization, are caused by changes in process or changes in manufacturing versus sample prep? And this is actually a very, very gentle sample prep. It's only 30 minutes in there. This is one of the advantages of having a QE instrument. You can do this workflow all on one instrument, but if you're a larger setup, then you can have multiple instruments and you don't need the QE and the GMP lab. You do a peptide map with top 5, you figure out which attributes to monitor, and then you pass the work off into the routine GMP compliance space. Chromeleon takes over LCMS data acquisition. It will do targeted peak detection, this is the first step of the MAM method. It's looking for the CQAs that you've decided are of importance that need to be monitored as part of in-process control or lot release. And then non-targeted peak detection. I call this the final sanity check. It's actually the final quality check. It's making sure that there's nothing unexpected. I think our former secretary, Don Rumsfeld, would say this is an unknown unknown. It's making sure that nothing that you weren't looking for suddenly appeared, that there's a change that you were not expecting to see that suddenly happened. An impurity, rapid change in one of the attributes, this will pick it up. It's kind of a blind final check. And this is all done based off of the work of Rich Rogers, who is the pioneer of this method. You can find the seminal paper here. If you need a copy, I'll get you one. So walking you through the method, this is kind of the dashboard that you'll get in BioPharma Finder. So the very first time, I have no idea what to expect. And BioPharma Finder is done a lot of the work for me to help figure out what I should be targeting in the first place. So you'll see a list of all of the modifications to the sequence. I think that's the laser here. And what's happened is we found an isomerization at residue 404. How does it know that it's this peptide sequence? Well, what it's done is it's used a kinetic model to predict not only which fragments you'll see, but also their relative intensities. So this is another layer of confidence. It's not only saying, yes, you'll see, Y12 will be the most intense at 2+. It's actually saying, not that you'll just see Y12, but you'll actually see it as the most intense. And if you look at the observed spectra that it's seeing here, it's actually a really good match to the theoretical fragmentation pattern. So I'm convinced that this is the peptide, but how do I know it's the ISO-D? It's actually looked and it said, well, I found a main peak here, and then I found a small satellite peak away from it. I'm expecting a retention time shift if it were an ISO-D of -1%. It sees it at -2% and says, yeah, this is probably an ISO-D. The peptide identity is confirmed from the MSMS spectra, and it has a retention time shift consistent with isomerization. Then it's done a lot of the work for me, which I like because I'm lazy, I guess. When you're developing this kind of assay, you need to know which charge states to look at, right? And this is a lot of method development work. You need to know which peptide should I target that's going to have the attribute. Should I target the miscleavage or the tryptic peptide? Which one is going to have the highest signal to noise? Which one is going to have the best intensity? Which one is going to be most reproducible? I don't know any of those questions a priori. BioPharma Finders actually said, hey, look, you want the +2 for this guy or the +3. He's the most intense. These are all the ones that I see, and the ones in blue are the recommended ones. And so now it's done a lot of the work to say, okay, I want the tryptic peptide, not the ragged. It's too low and too variable. It'll show you the CVs. It'll say you want the +3. And then it'll tell me which ions to target. And I can just put a check mark here and say, get me these. So right now, there's an intermediate step where you have to go through Pinpoint. This is advantageous because if you're already using Pinpoint for some of your workbooks, you can just directly import the Pinpoint work you've been doing directly into Chromeleon. This is kind of an undesirable step, so it's in beta right now. I have a beta version of this at the moment, so it will be released very, very soon. You can go directly from BioPharma Finder, which is the discovery phase, into Chromeleon. So you can say these components, which I think I want to target, they have the attributes I'm interested in, export check components directly to Chromeleon. And so it'll be a seamless interchange between discovery and routine. SR5 is out. I just wanted to touch base on this. It's a big performance boost. If any of you are using SR4, it's a free upgrade. SR5 is here. It's substantially faster if you're looking at a larger number of components. Generally, for lot release, we envisioned people looking at around like 20 to 30 different critical quality attributes, but a lot of people are using this in process, or they're starting with a very large list of attributes and then kind of weeding them out. And so this version of Chromeleon is a little bit more robust. There's also some additional data security and integrity. So for instance, as compared to, say, like a third-party, software, a vendor-neutral software, because Chromeleon can actually run the Q Exactive, if you don't take anything else out of this talk, that's it. Chromeleon can actually do GMP acquisition on an Exactive series instrument. Because it can do this, it can actually maintain a data vault, and it will run an integrity check to make sure that nobody has adulterated the data. So this means that nobody can swap in and out a raw file, rename it, cut it, slice it, say, you know what? This first injection kind of was bad, I want to swap it for another one. That's impossible. It will do a checksum on the data, and if the checksum doesn't match, it will put a red X and say this data has been tampered with, essentially, and won't integrate it. So what you'll see here, for those of you that are familiar with the Exactive series, here's the method editor. It's inside Chromeleon. Everything's the same, so you're not relearning, something completely different. It's the same setup. It's a very easy method. MAM is an MS1 only method. It's ideally suited for a GMP lab that's previously been using UV, right? So it's not a big transition to all of these gas phase fractionation methods or advanced MSMS methods like DIA. It's just very, very robust, very, very easy. And what you're doing here is you're taking advantage of the fact that you can access high resolution. The only tricky part here is you just set the resolution, and that's it. You tell the Q Exactive, do extra work, get me the highest quality data possible. But it doesn't make the method any more complicated. The LC method, pretty straightforward, the one I showed you earlier. There's a whole bunch of compliance tools. I get asked about this. You can turn them off or on, depending on whether you need to be GMP or not. You can have gated user access. So you can say, okay, only someone with credentials can log in, can actually calibrate the instrument, can actually tune the instrument. You have to supply this first before you can even access the Exactive tune. You can say who's allowed to electronically sign. So for instance, you can say I'm not allowed to electronically sign the report, but I can submit a report, and then my boss has to approve it. There's an audit trail, right? So you can see what's happened to the instrument, when, where. I have the tragedy in my life. I have to share my instrument with other people. This is very useful for figuring out what happened, when the instrument was calibrated last, who did it, what the result was, right? So it's very easy to see, okay, when was the last time somebody calibrated the mass accuracy or the analyzer accuracy? I can just go back and look through the audit trail. The other thing that you can check is there's also a data audit trail. So if you're still building your methods or changing your reports, you can see what's happened. So this guy, Mike Blank, he went in and he changed the scoring threshold. And it'll give you a red exclamation point, and you can see exactly what I did. I made this threshold a little bit more strict, and I'll show you an example of that. Calibration and tuning, all done through inside Chromeleon. It'll show you the last result. There is no last result, so somebody's in trouble. They didn't calibrate. We won't say who that was. Sequence setup, so then you set all of your injections, and all of the files that are associated with your injections stick together with the sequence, so it keeps everything organized. So this is one of the nice things about this MAM method, is that all of your processing settings, all of your report settings, all of your instrument settings, they're all kept together, well organized, and it'll associate everything with the sequence. So it's not just going through the Windows Explorer saying, oh, where did I leave that instrument method? Did I run this one, or did I run some other one? It's all here together. And you can put a comment, and it'll show you the version and what was run with what. Then you just walk down the list. Easy is good. Set the injections. Obviously, you need an instrument method to run the instrument. This is the targeted analysis, looking for the CQAs that are of interest. The final quality check, looking for anything new, and then you spit out a report. This can all be done automatically. Once you have the processing method set, and I'll show you that, all you have to do is actually click on the report, and it'll spit out a PDF. And that's it. You don't even need to go into the raw spectra at all. So you adjust the processing setting. I want to show you a couple of quick examples of how you might do this. So for instance, here you're going to set the, because you have everything in high resolution, you can set the mass integration tolerance. So this is going to be 5 ppm. You can put a moving average or not. You don't need to do any offline recalibration. There's no need to adjust your data, which is something that ideally you don't want. You know, for a regulatory agency, they may ask to see the raw data in its original form. You don't have to do an offline recalibration. You don't need to do a mass adjustment or anything like this. The data itself is good enough. You can do 5 ppm. And you can turn that smoothing off if you don't want it. 3-point smooth, I think I had 45 points across the peak. So a 3-point smooth is going to be a very, very negligible alpha. So you can just turn that off. MS settings, so okay. How do you know that you have the right peptide? First, you've identified it already. You have the BioPharma Finder results, which give you the MS spectra. They tell you the identity of the peptide. So you already know which peptide this is. But in an automated way, how do I make sure I'm integrating the right thing? So this is a great way to get a very easy green light, red light. Green light, I need to get at least 2 of the following. I need to get the isotopic dot product. What does that mean? It's a very fancy way of saying, if you have a peptide with a fixed chemical composition, right, it should have a fixed isotopic profile. How well does the isotopic profile match what you're actually seeing? The last is the mass accuracy, right? So the mass accuracy, that's not just of the deconvoluted or the monoisotopic mass, that's of all of the isotopes. Remember, we're at high resolution. So the mono, the M+1, the M+2, even the M+3, they all have to be within 5 ppm. It needs to find all of them as well. So you're not just integrating a spurious result where you happen to have something that matched the mono. It actually has to have the isotopic pattern, and they all need to be pretty good. And then the peak apex alignment, this is the alignment between the +2 and the +3 or the +3 and the +4 charge state to make sure that you really do have the peptide that you think it is. So for instance, like penny K shows up as a +2 and a +3 if you do trypsin. Are they eluting at the same time? Obviously they should be, right? So it checks that. You can set the individual integration settings here. So you can say, I want to do a split peak integration just for this peptide. Why would you need to do that? What happens if you have 2 structural isomers, say penny K, right? You could have deamidation on one or another, and there's actually a third asparagine as well. If it doesn't completely resolve all 3 distinctly, you'll have a split peak, right? How do you tell the software ahead of time, yeah, get me the left-hand feature, get me the right-hand feature? You can program this ahead of time to do it automatically. So for instance, the one where I usually do this is I have an ISO-D that likes to sit as a shoulder of the main peak. I can program in that this is a shoulder, and it will automatically find the shoulder peak. So I'm not sitting there having to reintegrate automatically all the time. And finally, the retention time settings. You set the retention time a priori and the integration, and you have a couple of options. You can set, relative as percentage of the retention time, or the first peak in the window, the greatest peak in the window, the one that's nearest to the retention time you set. Here's an example of how you can really, really automate this. Here's ISO-D from one of the stress samples that we have. So here's the main feature, and here's the ISO-D. It's around 20%. In the control sample, it's about 0.1%. How do I make the software not just pick this automatically? So if you go into like Skyline or Pinpoint, it's just going to pick the biggest one and grab this all the time. What you can do in this method for MAM is you can actually say, go find this one. He's at around 1E9. Find him, move over by minus 0.7 minutes, and then only look within plus or minus 0.5 minutes. And then what happens is, because he's always moving relative to the main feature, he can never accidentally integrate the main feature. So if you look in the integration window here, all he sees is the ISO-D. He can't even see this guy. If this one moves, it'll move this one as well. So this is a way that you can automate the selection of low abundance features without having to go through and manually reintegrate everything each time. And that's exactly what you want for a QC kind of environment. You want it to be able to do as much automated and robust and routine as possible. Double click and it gives you the report. Just to give you an idea, I think we're almost at the time limit. This is what the studio looks like. So you have your integrated ion, extracted ion chromatograms integrated here. The theoretical isotopic correlation, you can see the purple is the theory, and then the different colored lines are different injections. You can look at the raw data, of course. You get this green check mark, yes or no. Here's an example of deamidation, right? So in this case, what you look at Don't worry, take your time. What you're looking at here looks like a single peak, but I can tell you for a fact that there is deamidation. There is a deamidated peak in there. If you collect the data at low resolution, which is 30K, the formula M over delta M equals resolution means that your actual M over Z peak width is 40 ppm wide. So it is absolutely ludicrous to say I'm going to collect the data at 30K resolution and do quantitative integration at 5 ppm. You can't. The M over Z width is actually 40 ppm. That's how wide each one of those mono M+1, M+2 peaks are. 40 ppm. So I said, okay, let me try and do the extraction at 25 ppm. I do get two features, one for the main peak and one for the deamidation, but they have the same retention time, and I'm getting 72% relative abundance. This is obviously the wrong answer. Chromeleon is going crazy and spitting out a whole bunch of warnings. It's saying, take a look at this isotopic distribution. It's terrible. Why? Because I'm actually integrating the native peptide along with the deamidation. And so all of the isotopes are skewed off. I say, okay, let me try and force it at 5 ppm. It gives me a more reasonable relative abundance, but Chromeleon is still unhappy. Why? The peaks are misintegrated. You see, it's actually dropped the mono. Why? Because at 30K resolution, the M+1 of the native peptide and the mono of the deamidated peptide are within 5 ppm of each other. With a peak that's 40 ppm wide at 30K, they're just going to blend together. And you're going to get a mass that's neither one nor the other. When you collect the data at 120K resolution, and all you have to do is just put the checkbox on the exactive method, now you actually have a peak that has about 5 ppm of width. You can do the extraction, the integration, everybody's happy, you get the green check mark, that's what we like to see, and you get a reasonable relative abundance, about 0.25% for a freshly prepared sample. Finally, we have the non-targeted processing. So I'm going to skip these two in favor of this one. This one you have to click a bit. Okay, so we did a 2-week stress study at pH 8, right? So I'm not sure what to expect. The software will automatically go through and find new features that have changed. This has changed by about 7-fold. We generally recommend about a 10-fold change. The idea is to look for totally new features. The threshold that we set was about 1% of the tick. Remember, this is replacing a UV method where you look at 2 peptide maps by UV or and then you say, are there any differences between them, right? So mass spec we set to about 1%, and it found several new features. It found this guy. How do I know what he is? I go back to my original peptide mapping results. I look for this M over Z. It's penny K. It found this one. I'm like, okay, I wasn't expecting that. What is it? It's actually this iso D here. If I go back to the results, I'm like, really? It was able to pick up this iso D? And if you look at it, here's the raw data. Yeah, it did increase by quite a bit. So it didn't make a mistake and integrate the native peptide by accident or something like this. It found the little iso D and said, yeah, you know, this is changing, take a look. Then you can spit all of this out in a report. So here's the system suitability report. We run 3 technical replicates. We make sure that the retention time is stable. It'll automatically tell us pass, fail. Make sure that the peak area is roughly stable. I said like 20% injection to injection. This is the absolute peak area, not the relative abundance. We had a certain minimum threshold. We said, okay, if it drops below this threshold, I didn't prep the sample right. There's something wrong with the injector. Maybe the mass spec is losing sensitivity. We want to make sure that we get the minimum, and then mass accuracy, obviously. And this report generates automatically. It will reach into your sequence and grab any run that's been flagged with a system suitability run and pull it into this report. Here's another one for the CQAs. So here's a stress study. Here's penny K, went from about 1.5% across replicate digest to about 10% when you stress it out. ISO D, that was the one I showed you, 0.1 to 22%. Others were a little bit more subtle. This one went from 0.43 to about 0.8, so doubled after stress, but still pretty low. This one went from below the limit of detection, you can't see it in the control sample at all, to about 0.12%. So it's able to pick up big changes, small changes. It's overall consistent. This was a thermal stress of the sample, so deamidation and isomerization, of course, increased. Glycan profile, this didn't really change. between any of the replicate digests. I really wouldn't expect it to. We were able to see some low ones, like M7, he's in there at about 0.02%. So we got the major 13. You can look for more. There's a couple more in there. And then finally, all of this can be packaged up. So you say, okay, I have my processing method the way that I like, I have my report templates, I have everything that I like. I can package up the raw data, even the view settings, all of it, into a single CMBX file and transfer it to another installation. So I can pass this off to one of my colleagues. So for instance, if we have to do a demo, I hand them my CMBX file. They run the same product that I do, this NIST-MAB. My processing methods will work for them. They can just execute that and run the report. Maybe they change stuff if they use a different column, but that's pretty straightforward. And then to sum up, because I think I'm a little bit over. It's powerful. You can replace a lot of conventional lot release methods, but have deeper knowledge of the product. This has been used previously for filing to begin clinical trials with the FDA. So I'm not talking about something that could happen or might work. It was actually this software and this hardware, an Exactive Plus and a Q Exactive. Confident, so again, you have this automatic scoring algorithm. I think we extolled the virtues of high resolution. It's easy to use, it's simple, it's just an MS1 method. And I know to a lot of you that may seem like, oh, boring, whatever. But remember, this is designed to be something robust, automated, and be useful in a QC environment. So again, you're just saying, get me the highest quality data possible. You don't have to worry about any kind of advanced isolation or fragmentation methods. And then insightful, you can start to think about how to incorporate this into a quality by design regime. And that's all I had to say to you guys today. Thanks.